everybody. I'm Stephanie Hicks. I'm an assistant professor in the Department of Biostatistics at Johns Hopkins, and today I'm going to be talking about scalable statistical methods and software for single-cell data science. For those that are not familiar with single-cell RNA sequencing, I'm going to briefly cover the technology. We begin with a tissue sample, and we isolate and disassociate cells, for example, from a brain tumor, and we sequence those individual cells. We sequence the transcriptome of those cells. At the end of the sequencing experiment, we get a set of reads that map to a particular reference transcriptome. And so we count the number of reads, for example, that map to gene 1 in cell 1. We sum up those reads and we record them in a matrix. They're shown in the bottom left. If I did my math correctly, there are 18 reads in cell 1 and in gene 1. And we'd repeat this process for each gene and for each cell, recording the number of reads. And then using this matrix, we can do many different downstream analyses, such as look at heat maps of expression for a given set of cells in a given set of genes. And we can also apply things such as dimensionality reduction, such as principal components analysis, to look at the clusters of cells in our data, for example. So this is one comment that I often hear when people when people begin to use single-cell RNA sequencing data, and this is becoming more and more of a common comment. I have some awesome single-cell RNA sequencing data and I want to analyze it, but it is so big that it does not even load into memory. Help, what do I do? You may ask, how much of a problem is this? In fact, this is becoming quite a big problem. So here are 4 studies pulled from 2019. And I'm highlighting in red the number of cells that were sequenced in each one of these studies. And the point of this slide is to demonstrate that we are standardly producing single-cell RNA sequencing data sets with 1 million+ cells. So today I'm going to talk about 3 challenges related to scaling up single-cell data science. And the first is going to be focusing on scaling up clustering algorithms to millions or potentially billions of cells. So if we're going to talk about clustering, scalable clustering algorithms, what do we need to do this? Well, we're going to need to work with data that are either stored in memory, like we're all typically used to, or data that are stored what's called on disk. And the reason that we may need to work with data on disk is that, for example, if you cannot simply just load the data into memory, or you cannot perform some task such as clustering on the full data sets in memory. Some example file formats that you can use to store data on disk are HDF5 and more recently ZAR and TileDB. And I highlight that some of these formats have been adopted by global organizations such as the Human Cell Atlas. In addition to working with data on disk and in memory, we also want these algorithms to be fast. For example, we want to potentially cluster millions of cells in maybe a low-dimensional space, such as principal components analysis space. And maybe we want to do this multiple times. So for example, if we're using the clustering algorithm k-means, we would need to rerun this algorithm multiple times for different k. So let's talk about k-means clustering more specifically as the choice for unsupervised clustering here. I'm going to briefly introduce the algorithm if you're not familiar with it. For a given set of n data points x and a number of clusters k, the algorithm partitions the data into k clusters. It partitions the different observations into k clusters. And formally, this algorithm minimizes a within cluster sum of squares. In practice, however, the way we implement this algorithm is we use an iterative algorithm based on 2 steps, where for a given set of centroids, such as a random set of centroids, we assign each observation to the closest centroid. Given this label or this assignment step, we can then compute the new centroids for all the observations in each cluster. Now, one problem with k-means is that at each iteration of the algorithm, you have to have not only all the data stored into memory, but you also have to store all the pairwise distances between each observation and its closest centroid. So this makes k-means not scalable. In contrast, there's an algorithm called mini-batch k-means that was developed by Scully in 2010. And the idea here is that At each iteration, instead of using all the data in memory, we're going to only use a random subset of the data, and we call this mini batches. This could be a relative number, such as 1% or 5% of the data, or it could be an absolute number, such as 100 observations or cells or 500 observations in cells. The benefits are we do not need to store the whole data set in memory. We only need to store a subset of the data, specifically the choice of how much random data we want, such as 1% or 5%, and we call that the mini batch. And then also, of course, the k-centroids, but this is a very small amount of memory. And then at each iteration, only the distances between each mini batch and the k-centroids needs to be computed. This saves a huge amount of memory resources needed to be able to store these pairwise distances. And this also makes it a really natural candidate for clustering on-disk data because I only need to load in, for example, 1% of data at a time to be able to run this algorithm. I searched around for some existing implementations of mini-batch k-means. There is one in a package called cluster R within the R programming language. The problem with this package is that the input data must be in a base numeric matrix type. If you're not familiar with what that means, just know that these on-disk data formats that would be really nice to use are not possible to use with this package. So it does not make it a viable package for us. In contrast, there is a Python module, mini-batch k-means, in the scikit-learn module that does allow input from an HDF5 file, which is great. However, when you go to run the algorithm, it realizes the full matrix into memory, thereby not making it usable for us. And you can also use the mini-batch k-means function from Python inside of the reticulate package in R if you're interested. So we built a mini-batch k-means software package within the R and bioconductor ecosystem. And it is implementing specifically the mini-batch k-means algorithm for large data sets, and it also works with data that were stored in memory or data that are stored on disk. And our goal here was for anybody out there who's using the k-means algorithm would be to just simply replace k-means with mini-batch k-means, and this would enable a scalable version of a k-means algorithm by simply replacing one line of code in your package with this implementation of k-means. So next I want to demonstrate some of the advantages of mini-batch k-means. I'll state up front that mini-batch k-means is both memory efficient and fast. So we started with a data set with 1.3 million cells or observations, and we downsampled to data sets of sizes starting on the x-axis of 75,000 to 1 million observations or cells. And on the y-axis, I'm showing you the max memory used in figure A and the elapsed time in minutes in figure B. And the three algorithms I'm comparing are k-means in red, mini-batch k-means with data stored in memory, and mini-batch k-means with data stored on disk using an HDF5 file. And what you see is k-means is in fact very memory inefficient as the data sets get larger, and it also takes a takes a large amount of time to run as the data set gets larger. In contrast, if you just simply replace mini-batch k-means with data that are stored in memory, you get savings in not only the memory used, but also significant savings in time to run the algorithm. And if you're willing to store the data on disk, such as an HDF5 file, and use mini-batch k-means, you can cluster 1 million cells with less than 2 gigabytes of memory in under 10 minutes, which is really quite phenomenal. In addition to this, mini-batch k-means is as accurate as k-means for batch sizes of 500 observations or more. So I introduced the concept of mini batches in an earlier set of slides, and here I'm showing you absolute batch sizes. So batch sizes of ranging from 250 to 500 to 750 to 1,000. So these are the number of cells that I'm loading into memory at any point of the algorithm. And I'm showing you this for data sets of 5,000, 10,000, and 25,000 going from left to right. On the y-axis, I'm showing you the within cluster sum of squares criterion, which is classically used to measure accuracy. And what we see is K-means does not change, the accuracy does not change as a function of the batch size, which is what we expect. However, mini-batch K-means and both data stored on disk and in memory become as accurate as K-means for batch sizes of around 500. So this is great. We also applied mini-batch K-means to the 1.3 million cells that I talked about before. On the left is the UMAP representation of the 1.3 million cells, colored by the predicted label for each cluster using mini-batch K-means. What we did was we took our 1.3 million cells, we performed principal components analysis, we extracted the top 50 PCs, and performed mini-batch K-means 16 different times to look at the different K. And this process took a total of 3 minutes. We also compared this to the state-of-the-art SNN, shared nearest neighbors, plus Louvain clustering within the bioconductor framework, and BBKN with Leiden clustering within the ScanP framework. And these algorithms took a total of both 35 minutes and 48 minutes, respectively, thereby demonstrating significant savings in time using mini-batch K-means. If you want to know more about our package, I encourage you to check out our publication in PLOS Computational Biology. All right. So the second challenge I'm going to focus on is scaling up education for the analysis of single-cell data. This is another comment that I often hear when somebody starts to analyze single-cell RNA-seq data for the first time. They're often overwhelmed and unsure kind of where to begin. And they say, I really don't know what I'm doing or where to begin. So I wrote a paper to try and address this problem. Specifically, my goal was to help users and researchers navigate the world of single cell within the bioconductor ecosystem. In addition to this paper, the part that I'm most proud of is an online book called Orchestrating Single Cell Analysis within Bioconductor. It's written in Bookdown within R Markdown. And it consists a set of 42 chapters, including 18 complete workflows, starting from data import to data summarization, walking you through step-by-step how to analyze your data using the bioconductor ecosystem of packages for single-cell data analysis. There are lots of philosophical discussions about single-cell analyses, such as a classic one that I often get, which is, what is the true clustering in my data? And most importantly, it includes lots of code for users to simply copy and paste to be able to analyze their own single-cell RNA-seq data. The third challenge that I'm going to talk about is specifically spatially resolved transcriptomics. And here I'm thinking about scaling up single-cell data science in terms of how can we extend the methods that were developed for single-cell to this exciting new technology. Now, these technologies, spatially resolved transcriptomics, are truly hitting their stride right now. For example, you might have heard of MERFISH or SlideSeq or 10x Genomics Visium. Nature Methods actually labeled spatially resolved transcriptomics as method of the year. So now, if we think about looking at an organ, such as the brain here, shown in Legos, if we think back through all the technologies that we have been using to date to explore the transcriptome of this organ, such as bulk RNA sequencing, single-cell RNA sequencing, and now spatially resolved transcriptomics, we can start to look at the gene expression in the original tissue itself, which is really exciting. Now, of course, with these technologies, these new technologies comes exciting opportunities or problems that we have to deal with. So in my group, we began with asking a really basic question. Given this new technology, what is the best way to store this new data type? And this is kind of like stating we need to learn to crawl before we can walk. Spatially resolved transcriptomics data is more complex than just single-cell gene expression data because in addition to the transcriptome-wide gene expression data, we now also have this 2D spatial coordinates that we have to think about with these XY coordinates. And we potentially have some kind of image data that we would like to integrate with these data. So to address this, my group, in collaboration with researchers from the University of Padua and University of Zurich, we developed the Spatial Experiment class within Bioconductor. In addition to not only providing a standardized data infrastructure, it also enables modularity and interoperability of packages that adopt this class, which is truly exciting. It can store both spot-based data, for example, 10x Genomics Visium platform or SlideSeq, but also molecule-based data, for example, SeqFish and MERFish. And this work was led by 3 researchers, Daria Righelli, Helena Kroll, and Lucas Weber. Lucas is in my group. There's a preprint available online if you're interested in learning more about it. Okay, so now that we've developed a robust and standardized way to store spatially resolved transcriptomics data, we can start to effectively analyze these data in a really reproducible way. And to do that, we had to start someplace that we're comfortable with and that we know is well studied. So using a technology called the 10x Genomics Visium platform, a team of collaborators at the Lieber Institute for Brain Development and a set of researchers at 10x Genomics and a postdoc in my group, Lucas Weber, we recently profiled the human postmortem dorsolateral prefrontal cortex tissue sections from 2 pairs of spatial replicates from 3 independent neurotypical donors. So you see there's these 3 donors here, and we created a data set with 12 different 10x Visium data sets, but these come with little replicates that were right next to each other. And you can see that what we're capturing here is to look at both the white matter, but then layers 1 through 6. If you want to look at an example of what the data look like, here's a global view of the 12 sections showing the gene expression of PCP4. And this is a marker gene that is highly expressed in layer 5 of this particular tissue. And this protein is important for synaptic plasticity. So using a combination of histology, graph-based clustering, and previously identified layer markers, we manually annotated the 6 layers of the cortex. They're shown in the top left for each of the 12 tissue sections. And we did this in a Shiny application. And effectively what we're doing is we created a ground truth label for each spot, knowing whether it's in the white matter or layers 1 through 6. Then we compress the spot level data to layer level by aggregating across the spots to create a pseudo bulked counts matrix. And when we applied principal components analysis to these 12 samples for these pseudo bulked count data, we found that PC1 explains 34% of the variance, differentiating mostly between white matter and layers 1 through 6, while PC2 explains 9% of the variance, differentiating between layers 1 through layer 6, which was really exciting to see. Now, we've made these data available to download via Bioconductor, and you can also, as I mentioned, explore these data in a Shiny application. And this has made it really easy for other authors to use our data in the evaluation of methods that they're developing. So for example, the authors of a data-driven clustering algorithm called BaseSpace used our data to compare their data-driven clustering method to existing clustering methods, including methods that were developed for both SingleCell and spatial methods, such as Giotto and ST-Learn. So I just want to say thank you to all of my collaborators in the Orchestrating SingleCell Bioconductor project, mini-batch k-means, the 10x Visium dorsolateral prefrontal cortex project, and the spatial experiment class. Thank you very much to the generous funding support, and I will stop there and take questions.